Bismillahirrahmanirrahim. This is Dr. Nighat Aslam from Department of Biochemistry, Independent Medical College, Faisalabad. Today we are going to start our new topic, transcription, and this is first lecture. Genetic master plan of an organism is present in the DNA, and this DNA is expressed in the form of RNA and proteins. RNA are formed, molecules of RNA, they are formed by a process known as transcription. RNA and protein, they are the express product of the DNA, and it is the RNA which is the first working copy of the DNA. The final product of the DNA, it can be the protein or it can be the RNA. It depends upon the gene. About 2% of the genome, it encodes proteins. Rather, it is less than 2%. Other part of the genome, that is more than 98% of the genome, is introns. They may be the untranslated regions of the messenger RNA. They may be the regulatory DNA sequences or some mobile genetic elements, but they do not code any of the proteins. So the transcription is a process which involves the enzyme DNA-dependent RNA polymerase and a number of associated proteins. General steps involve initiation, elongation, and termination. The DNA regions which are located upstream, and the protein factors that bind to these sequences to regulate the initial transcription, initiation of the transcription, DNA regions are the promoter regions, and they are well known. This initiation step, or the proteins and the part of the DNA which is involved in the initiation, it is well established. mRNA in particular have very different lifespans in cell. It depends upon the need of the mRNA, how much protein is needed and how much time that protein need to be active. Transcription is highly selective, mean not all of the DNA it is transcribed. As I have told you before, RNA may undergo modifications, and that modification change the RNA into functional molecule. Before that modification, the RNA cannot work. Modulation process results in altered rate of protein synthesis, and thus a variety of both metabolic and phenotypic changes occurs. And this is how the organisms they adapt to their environment, and this is how the cell they are differentiated. Errors or change in any of the above process can lead to serious diseases. These are the types of the DNA. Types of the DNA, it can be divided into RNA can be divided into two main parts. One is the coding RNAs, protein coding RNAs. These are the mRNA, and non-protein coding RNAs. And non-protein coding RNAs, they may be the large non-coding RNAs and small non-coding RNAs. Non-protein coding RNAs contain ribosomal RNA, long non-coding RNAs, transfer RNAs, small nuclear RNA, and micro and silencing RNAs. These are the two types of the RNAs in prokaryotes and eukaryotes, and two species of the RNA prokaryotic ribosomal RNA and eukaryotic. In the prokaryotes, there are three species of the ribosomal RNA: 23S, 16S, and 5S. In eukaryotic ribosomal RNAs, they contain 28S, 18S, 5.8S, and 5S. These ribosomal RNAs, they are present in association with proteins. This is the diagram of transfer RNA, which is of clover shape, and it contains intrachain bonding and a lot of and many altered bases present in the transfer RNA. Their structure of all the RNAs you have studied in first year also. This is the mRNA, which contains the 5' cap and 3' poly-A tail, and this is the mRNA which can be or which are translated into the proteins. RNA synthesis has some points which are similar to the DNA synthesis. It has the same initiation, elongation, and termination points, and the RNA is synthesized with the same polarity, that is from the 5' end to the 3' end. RNA synthesis also occur starting with its 5' end and towards the 3' end. Large multi-component initiation complex, they are present during the polymerization, and Watson and Crick base pairing rules, they are also there. The differences is that ribonucleotides, they are used as a substrate for the RNA synthesis rather than the deoxyribonucleotides. In the DNA, there is the thymine, but in the RNA, U replaces thymine. Primers are not involved in RNA synthesis. Mean primer is not required for the initiation step for the synthesis of RNA. Only portion of the genome are vigorously transcribed or copied into the RNA, whereas entire genome must be copied once and only once during the DNA replication. It means only portion of genome it is transcribed into the RNA, but in the DNA replication, whole of the chromosome, whole of the genome, it must be copied once and only once, but a number of RNAs, they can be formed. So this is also one of the difference. No highly active efficient proofreading function during the RNA transcription. This is DNA strand. This picture shows that any of the two strands, they can be served as a template strand for the RNA synthesis. Here you are seeing that there are the 4 genes, gene A, B, C, and D. In A, B, and D genes, the lower strand, it is serving as a template strand, and this strand is read in the direction of 3' to 5' direction, but the protein will be synthesized, RNA will be synthesized in 5' to 3' direction. And this gene C, the upper strand, which here it is also shown that the gene is transcribed in 5' to 3' direction, and the template, it is read in 5 to 3' directions. So any of the two strands, they can be served as a template strand. The strand which serve as a template strand is the non-coding strand, and the other strand which is copied, which is 100% copied, is the coding strand. And this is the nascent strand, which is similar, which has the sequence similarity with the coding strand. So the template strand is also called the non-coding strand, The template strand is also called the antisense strand, and it is also called the minus strand. This is the enzyme DNA dependent RNA polymerase, which has the different subunits 2 alpha, 1 beta prime, and 1 sigma. This protein, it is 400 kilodalton, and its size is 400 kilodalton, and E enzyme associated with sigma to form the holoenzyme. This is the different, these are the different functions of the subunits of the RNA polymerase. The 2 alphas, these 2 alpha, they are required for the enzyme assembly. Also it is written in several books that this sigma this omega subunit, it is also involved in enzyme assembly. The template binding function is present in the beta prime, and polymerization activity is the function of beta subunit. Sigma factor, it recognizes the promoter region. This sigma factor, it recognizes and it is required for the recognition of promoter region. Transcription is can be explained in a cyclic manner. In the cyclic form, it can be described in 6 steps. The first step is the template binding and close complex formation is formed. Close complex formation is formed when the RNA polymerase, this yellow colored circle or yellow colored ball, this is the RNA polymerase. It binds at the promoter region, and close complex formation is formed. The second step is the open complex formation, in which the RNA polymerase, it moves, and at a part of the DNA, it melts. RNA polymerase melt the 2 DNA strands, and after melting, it is called the open complex formation. It is also referred to as pre-initiation complex. Pre-initiation complex. At this stage, PIC also formed. Then is the next step, chain initiation. Chain initiation, in which the RNA polymerase catalyze the coupling of the first base to the second base and form the dinucleotide. Here you are seeing that dinucleotide, it is formed. And now is the step which is called the promoter clearance. Mean the RNA polymerase, it has been moved towards the gene, and the promoter region, it has been cleared, it has moved further. And NTPs, these NTPs, they are used as the substrates. About 10 to 20 nucleotides of the polymerization it has occurred, and here the conformational change also occurs in the RNA polymerase, and the sigma factor, it is released. Now is the function of only core enzyme. Then is the chain elongation. In the chain elongation, upon encountering the transcription, in the chain elongation, further NTPs, they are added, and they are added at the 3' end of the RNA, and this is the RNA transcript which is elongated, and it will continues doing it will continue to doing its work, and until there is the signal present in the DNA, and the RNA polymerase, it will stop its function, and here the chain termination, and in the chain termination the nascent RNA, it is released, RNA polymerase is released, and this DNA, it is cleared. Now for another cycle, or for an production of another transcript, RNA P will again bound, and this transcription cycle will continue until our required RNA products, they are formed. In initiation step, first RNA polymerase holoenzyme, means enzyme and plus the sigma factor, it bind to the promoter. Once the promoter is located by RNA polymerase, enzyme promoter complex, DNA complex, undergo a temperature dependent conformational change and unwinds at transcription start site +1. The sigma subunit enable the core enzyme to recognize and bind the promoter region. I have told you before that the sigma subunit of RNA polymerase has the function to recognize and bind the promoter, and here the pre-initiation complex is formed. Unwinding allows the active site of the enzyme to access the template strand. This is the DNA, which shows that the two promoter regions, they are required in the prokaryotic initiation transcription of the initiation of the transcription of prokaryotic genes. You are seeing that there are two promoter regions, -35 region and -10 region. This -35 region, it is a consensus sequence, and promoter regions, all the promoter regions, they are the consensus sequence. I will tell you in the next slide that how can you define the consensus sequence. Consensus sequence means the sequences are always the consensus sequences are the sequences in which the base shown at each position is the base most frequently encountered at that position, means their sequences are always same. This -35 region is the initial point of the contact for the holoenzyme, and these promoter regions, they are present at the upstream of the transcription start point. This is the transcription start point. Upstream means that the sequences which are towards the 5' end of the gene, these are the upstream region. This is the upstream region towards the 5' end of the gene, and the regions which are towards the 3' end, they are the downstream regions. This is the transcribed region. And the second promoter region is the Tata sequence, or it is also called the Pribno box. Holoenzyme, when clear or move forward towards the Tata region, and this is the second consensus sequence, and it is located at the -10 region, means the 10 nucleotides before the transcription start point. Here, this is the point where the closed initiation complex is changed to open complex, and also the transcription bubble is formed. Promoter regions, they are the -35, no box, which is also called the data region, and transcription start site or TSS, it is the +1 site I have shown you before. And the RNA product which is synthesized in the 5' to 3' direction is the primary transcript, or simply we can use the word transcript for the RNA molecule. Transcription unit, this is the term which is often used in genetics. What is transcription unit? It is the sequence of the DNA, or it is the part of the DNA which code for a single RNA molecule along with the sequences necessary for its transcription, normally which contains the promoter and RNA coding sequences and a terminator. So all this is a transcription unit. This picture shows the transcription unit, which contain the promoter region, which contain the gene, and which also contain the termination signals. This is the transcriptional unit. Approximately 35 base pair upstream of the transcription site, there is a consensus sequence of 8 nucleotide pairs, and to which RNA polymerase binds to form the so-called close complex. So the first, when the RNA polymerase bind at the first promoter region, which is at -35 base pairs, the complex will called close complex. And when it move forward and there is this strand separation, the combination of RNA polymerase plus the promoter then form the open complex. Now come to the elongation step. The elongation phase is said to begin when the transcript exceeds 10 nucleotides in length. At this stage, holoenzyme do not work. The sigma part of the RNA polymerase, it will separate it out. There is the conformational change in the RNA polymerase, and the core enzyme now works. It uses, in the elongation step, it uses nucleoside triphosphate as a substrate and releases pyrophosphate, PPI. Enzyme works in processive manner, it do not require primer and no proofreading activity. But misincorporation of ribonucleotide causes RNA polymerase to pause, backtrack, cleave the transcript and then restart. So it means that there is some proofreading activity, but this is not in such a efficient manner which was present in the DNA replication. So transcription has a high error rate than does replication. In the elongation step, DNA unwinding, it can cause the supercoiling, which is removed by RNA topoisomerase enzymes, right? And RNA polymerase also has intrinsic unwindase activity that opens the DNA helix. DNA unwinding occurs at a rate of 20 base pair per polymerase molecules. This is the diagram which shows that RNA polymerase forms the transcription bubble. This is the RNA polymerase, and the DNA is open up here. This is the template strand, this is the template strand, and this is the RNA transcript which is formed in 5' to 3' direction, and these are the positive supercoils which are formed ahead of the replication fork, and these are the negative supercoils which are formed behind the supercoils. These supercoils will be removed by the topoisomerase, and the RNA polymerase will move further in order to complete the transcript. Now the termination. Termination can be of two types. It may be the row independent. Row is a protein. If it is not involved in the termination step, then it is the row independent termination. In row independent termination, which is seen in most prokaryotic genes, it requires the sequence in the DNA that is self-complementary. These two sequences, they are self-complementary, and when the RNA will transcribe this region, it will form intrachain bonding. They fold over itself and a hairpin structure is formed. And this hairpin, it will hinder the work of RNA polymerase, and RNA polymerase cannot move further. Also, the RNA transcript, it contains U's at the 3' end, and this bonding of U's to the A, there will be the weak bonding, and at this point the RNA polymerase, it cannot move further, and this transcript, the newly synthesized RNA, it will be separated, and there will be the termination of transcription. This is the row. Second type of the termination is the row dependent termination. Row is a hexameric ATPase with helicase activity. It contains ATPase and helicase activity. The ATP dependent helicase activity of the row separates the RNA DNA hybrid helix, causing the release of the RNA. Diagrammatically, it can be shown here, like that. This is the row protein. It is a hexameric protein, and it will bind at C-rich row utilization site, RUT, row utilization site, and near the 5' end of the nascent RNA. Then, using its ATPase activity, moves along the RNA until this row reaches the RNA polymerase, and this row causes the RNA polymerase to stop its working, and there will be the termination. The ATP dependent helicase activity of the row separates the RNA DNA hybrid helix and causing the release of the RNA. So the RNA polymerase, this row and transcript, they are separated out, and there is the termination of transcription, which is the row dependent termination. What are the actions of antibiotics in the transcription? Rifampin. This is a drug which is used in the treatment of tuberculosis. This drug, rifampin, it inhibits the initiation of the transcription. It prevents the chain extension beyond the 3 nucleotide, and it is I have told you before that it is used in the tuberculosis. Dactinomycin is used in the cancer chemotherapy. It interferes with the movement of the RNA polymerase along the DNA. It intercalates in the grooves of the DNA. Dactinomycin intercalates in the grooves of the DNA and interfere with the movement of RNA polymerase. So this is all about the transcription in the transcription of prokaryotic genes. In the next lecture we will start with the eukaryotic gene transcription. So best of luck. Allah Hafiz.